Hi, we are the Genetic Regulatory Council reviewing the CRISPR-Cas9 gene therapy proposed for achondroplasia. Achondroplasia is essentially the medical term for dwarfism, and it is a result of cartilage turning into bone during fetal development, particularly in the arms and the legs. Both males and females with this disease are short in stature, approximately 4'1", 4'4". Additional characteristics that come alongside the disease are macrocephaly, a prominent forehead, short upper arms and thighs, and fingers that merge together. And secondhand effects include, but aren't limited to, frequent ear infections, obesity, and apnea where sleep slows to a stop. Achondroplasia occurs in about 1 in 15,000 to 40,000 newborns in the United States annually. Achondroplasia is inherited by an autosomal dominant pattern, meaning only one parent needs to have the dominant gene for the child to present with achondroplasia. However, most patients with the mutated gene have genetically normal parents, meaning that it is a new mutation recurring after fertilization. And given the manner that it's passed down between generations, patients with achondroplasia have a 50% chance of passing their mutation and its consequences to their children.
So the FGFR3 gene encodes for the synthesis of fibroblast growth factor receptor 3 protein. There are 4 fibroblast growth factor receptors that have various different functions, including regulation of cell growth and division, determination of cell type, formation of blood vessels, wound healing, and embryo development. Mutations in this gene can lead to many disorders, including bone and skin growth. The change in formation of FGFR3 gene causes the FGFR3 protein to be overly active. FGFR3 is a negative controller of bone growth, so overactive FGFR3 affects skeletal development and disturbs bone growth, specifically problems in converting cartilage to bone. So this results in achondroplasia characteristic short arms and legs.
So just a quick overview of CRISPR-Cas9 technology that would be used in this situation. Cas9 is an enzyme complex that takes a guide RNA and finds a specific DNA section to pair with. The DNA section is the one that you were trying to splice out. And once the guide RNA matches perfectly with the DNA strand, the Cas9 enzyme complex cuts the strand on both sides, removing the mutated gene, and then homology-directed repair inserts new nucleotides to replace the mutated gene with a properly functioning one.
Current therapies. Current treatments for achondroplasia are focused on alleviating complications, such as breathing difficulties arising from abnormal spinal growth or ear infections. Some patients have been treated with growth hormones, however, incremental height is minimal. BioMarin's Voxzogo has just received FDA approval to increase linear growth in children with open growth plates. Voxzogo binds to natriuretic peptide receptor B, which is a positive bone growth controller and stimulates bone growth. One of the side effects of lower blood pressure may be of concern.
The proposed therapy is from Genetis Inc., which is a Tokyo-based conglomerate that has proposed the use of CRISPR technologies to edit a patient's FGFR3 gene in order to allow them to not have the effects of the disease, but also to be able to pass on properly formed genes to their children. So the editing is done on germline cells. The solution, they're proposing a solution to a serious issue, and they're utilizing modern gene therapeutics to prevent achondroplasia. They start by isolating germline cells, sperm, and ova, and they use CRISPR-Cas9 technology to remove the mutated gene and reinsert a healthy gene in its place. Then they fertilize the altered germline cell, and so healthy genes are passed to the offspring, as well as allowing a normally functioning child to be born.
The FGFR3 gene is located within chromosome 4 of the 23 chromosomes and contains instructions to make FGFR3 protein. The sequence of amino acids on FGFR3 protein is determined by a specific sequence of 3 nucleotides in the gene. Nucleotides for position 380 on the protein chain should be GGA or GGG, which encodes for the amino acid glycine. Achondroplasia is caused by a mutation in this nucleotide sequence at position 380. So CRISPR-Cas9 is the technology being used to gene edit. CRISPR-Cas9 is used to identify the specific nucleotides for position 380 in the FGFR3 gene that is mutated. The Cas9 unwinds the mutated DNA and uses a guide RNA to search the mutated DNA for complementary spacer regions. Once this specific mutated region of DNA is found at the nucleotides for position 380, the Cas9 cuts the DNA and uses a DNA template with the CRISPR machinery to insert a copy of the correct code of nucleotides, which should be GGA or GGG.
This slide goes over why FGFR3 editing is necessary and ethical to treat the condition at hand. Number one, androplasia is not otherwise preventable, predictable, or curable. During pregnancy, the mutation in this gene escapes pre-implementation genetic diagnosis, and it can happen unexpectedly during pregnancy. It is also not otherwise curable post-birth. Therefore, any efforts at prevention is worth pursuing. Number three, another consideration is that replacing a mutated FGFR3 gene with a healthy one does not have other unforeseeable side effects, because the only function of this gene is to slow down the formation of bones by inhibiting the proliferation of chondrocytes. FGFR3 is the only known gene associated with achondroplasia. Therefore, gene editing has a high success rate. Regarding the ethical considerations, we believe that the FGFR3 editing doesn't suggest genetic superiority because of the long-limbed, because short-limbed dwarfism is not primarily an aesthetic issue, but a functional issue, because it largely limits the mobility of the patients affected. And lastly, since an informed consent is inherently impossible to obtain with germline gene editing, we believe this is a negligible issue because it's safe to assume that dwarfism is not favorable to anyone.
All that being said, there are some weaknesses and risks that we have to be mindful of. The first one is that we don't know the long-term effects that the gene editing by CRISPR has on humans yet, because there haven't been enough clinical trials done. We would need to observe many generations in order to be fully aware of the long-term health impacts, and if there are any that are passed down. So we would need to conduct some prospective clinical trials that would go on for a long period of time in order to be aware of those. And then second, there's also a risk of immunotoxicity, and some studies have shown that some individuals carry an anti-Cas9 antibody, which means that they actually have immunity against the commonly used bacterial nucleases in the technology, which can be an issue. And then also the large size of Cas9 creates difficulty when delivering cells via vectors. There are also limited target sequences that can be modified due to the need of PAM sequences. And fifth, the Cas9 might actually cut the DNA at the wrong sequence, leading to different health issues. And this would occur if there is a sequence that is very similar to the target sequence, and the enzyme would by accidentally cut at the similar sequence, and this could just cause some health issues like the development of cancer. And lastly, this technology could also increase the risk of cancer by selecting p53 deficient cells, which would result in mutagenesis and then tumor growth.
Ultimately, our decision is to approve this technology for commercial use for the following considerations. Achondroplasia severely worsens the patient's quality of life. Therefore, pre-birth prevention would be very helpful if it is indeed proven to have high success rate. Scientifically, achondroplasia is unpreventable and untreatable without germline gene editing of FGFR3 gene, and ethically, the editing of this gene will not promulgate genetic superiority because its goal is medical and therapeutic in nature instead of aesthetic.
So considering the lack of the potential side effects and the relative certainty of the success of this technology, and its ability to improve the quality of life, we believe that the gene editing of FGFR3 is worth pursuing without patient consent, which is impossible to obtain just because it would increase the success and the quality of life of that individual. Our next step is to fairly price the treatment by inviting its inventor, geneticists, and industry experts to discuss it. and here are our sources. Thank you.